contribution to the field and for coming for this talk. So, Luciano will talk about the importance and influence of environmental microbiota on biofilm formation. Thanks, Luciano.
Everyone. First of all, I'd like to thank the organization of the IAFP Latino and of course Dr. Valentina Trinetta for inviting me to take part of this roundtable. It's a pleasure to talk about biofilm because in my opinion biofilm is the most important topic for food safety, and I have been working with biofilm for the last decade. So I'm excited to talk with you today, but I have some problems with my English. Sometimes I make confusion with prepositions, but maybe everything's okay. And let me see what else. So the title of my presentation is The Importance and the Influence of Environmental Microbiota on Biofilm Formation. In my first slide, I brought here a picture for use for example. Imagine if I ask you, is the surface okay in your opinion? If I tell you this surface was after SSOP procedure or pre-operational hygiene process, maybe we have some dirt material here in the surface. We can see maybe a lot of blood or organic material. So they have a problem with their SSOP. And I have a second question. They have a problem with biofilms? Of course they have, of course. But they need to change the SSOP procedure first, because they have a big problem with their hygiene. If I show you the same picture, now everything seems clean. And we don't see anymore the dirt material. Now, they continue with the problem, maybe with biofilms, but they don't have, with a naked eye vision, they don't have this problem anymore. But it's time to the people of quality control started to thinking about to control the biofilm. And how? Sometimes they use the surface to check the contamination there. And check how? Using a cotton swab to take some samples on the surface to check the presence of the pathogens, for example, or some group of bacteria, for example, indicators bacteria, zoophilics or coliforms, etc. But we have two problems here. Sometimes we choose a place that the pathogen is not in the right place that we try to find the problem. And the result can be negative, but the pathogen is there. For example, Listeria. And in my opinion, the second problem is the choose of who I try to find. Listeria, Salmonella, E. coli, others. So I needed to check one different kind of protocol to find the contamination. So if I try to find Listeria, for example, I need to use a protocol specific for Listeria. And we never thinking about using different approach, for example, to have information about the other microorganisms, for example, by study of microbiota on the surface. Maybe it's an interesting approach. So now, because in this diagram you can see that the biofilm on surface, there is a big community with a lot of different kinds of bacteria, virus, molds, yeasts, not only one genera or one species. So sometimes the pathogen is not there inside this biofilm, but the microbiota is very big, and there is a lot of different kinds of microorganisms, and it's not possible to find the pathogen. And maybe we can change the way to find the problem. I will show you this Malu dissertation entitled Distribution, Adhesion, Virulence, and Antibiotic Resistance of Persistent Listeria in a Pig Slaughterhouse in Brazil. In this work, we have found more than 50% of all conveyor belts contaminated with listeria, and other places too. And this contamination started in 2016 and continued until 2017. This contamination was in a period of the time. And we have a very interesting information here that all the isolates of Listeria monocytogenes here in the surface was the same. Sent at 100% of similarity using PFGA analysis. And imagine what happened with the contamination of the food during this period of the time. The pork meat, for example, in this industry. And this was the conveyor belt that we work. We can see here in the naked eye vision, full of marks and grooves, allowing the listeria stay there for ages. And the most interesting thing that the industry, the people of quality control, they didn't have information about the contamination. They have a lot of lab analysis, and the listeria, zero. And we stay there for a period of time, and we analyze a lot of samples, and we have found more than 50% of contamination. After that, we did a lot of different works, different research with biofilms, and I will show you these two of Leonardo thesis. And it's a kind of new approach, not a new approach, but a new approach for us in our lab, in our team, to use a different way to find information about the surface and their contamination. I will show you this first paper published in 2023. The title was Pre-Operational Cleaning Process Interfere with Microbiology and Presence of Listeria and Salmonella on Food Conveyor Belts of a Poultry Slaughterhouse in Brazil. So we did a method, a phenotypic method to find these two bacteria, Salmonella and Listeria. And here you can see that the Salmonella contamination reduce, and we can find the elimination of Salmonella after the cleaning process, after the pre-operational cleaning process. But with Listeria, we have a significant reduction, but we have their persistence after process of cleaning. So maybe this Listeria was there, for example, like a biofilm. So regarding our results, with bacterial genera identified before and after pre-operational cleaning process using 16S RNA, we have found a lot of different genera and species. I will show only the genera. The Pseudomonas acinetobacter and Aeromona was the most important after process of cleaning. I will show two more information here. If you look at only the colors here in this picture, you can see the change, the colors before and after. So if the colors are changing, the bacteria are changing too. And we have to look at only after cleaning because it's more important for us. And we have another information here. We need to recognize that after a cleaning process using in the industry, with the microorganisms don't vanish, they stay in the surfaces. We read a lot of papers and the results was the same, but we need to prove in our lab this occurrence too. Here, another way to see the same results. Here we are looking at beta diversity of microbial ecology according to the sanitization process. Here in this PCOA graphic, you can see two distinct groups using both methods. Here in red color, you can see the samples collected before process of hygiene. And the blue color, we can see all the samples was collected after process of cleaning. And we can conclude that the number of microorganisms and the abundance, relative abundance of the microorganisms were different. So they had an influence by the process of hygiene. The process of hygiene changes the microorganisms that live on that surface. And here we have a third way to see the same data using the three groups of bacteria. The first group in red is the groups, genera, and species that we found before the process of cleaning. The second group were composed by microorganisms after process of hygiene. And the third and maybe more important group was that we find microorganisms in both moments. They are on the surface after and before. Maybe this group of microorganisms, they are tolerant to the process of hygiene. And consequently, maybe they are in a form of biofilm, in a possibility to form biofilms on that surface. And we conclude in this study that salmonella elimination after cleaning process and disinfection. We proved to the persistence of listeria even after process of hygiene. The persistence of microbiota, Pseudomonas, Aeromonas, etc. And maybe they can be tolerant to the hygiene process and form biofilms. And we saw a great diversity of culture between the surfaces before and after. And the second research of the thesis was characterization of microbial ecology Listeria salmonella on equipments and utensils surfaces in Brazilian pork, poultry, and dairy. The difference between this work, we try to compare the microbiota in three different industries here. We don't have enough time to see all our results. I will show you only the Venn diagram here and only three information using this Venn diagram. The first information is, I don't know if you see, but maybe you can see, but you believe in me. And Pseudomonas on poultry, on pork, genera and species, and Pseudomonas putida, and Pseudomonas putida. Pseudomonas putida we have found in all three different industry. So maybe this information is important because Pseudomonas belong to the microbiota inhabiting these three facilities. And maybe their characteristic, the psychotrophic characteristics of Pseudomonas maybe contributed to the results, but because all the room's temperature that we make our samples there was below the 10 degrees Celsius. So maybe it was important variable to control, to select the bacteria. The second information is poultry and pork, these industries sharing same genera, but when you see the genera and species in their industry, with exception Pseudomonas, other bacteria are not the same. For example, Curia, Staphylococcus, we have found only in the dairy industry. So we conclude in this study that the type of industry influences the microbiota diversity. Pseudomonas was, we have found all evaluated environments. Poultry and pork, they have similar microbiota, but dairy not similar, was different. So maybe a more conclusion, a more widespread conclusion, we can see that Pseudomonas, Acinetobacter, Aeromonas and other, maybe they have a great influence on the surface of industry, mainly after pre-operational hygiene process. The contamination was similar at poultry, pork and milk because of the Pseudomonas. They have all this microbiota, have a potential to produce biofilms. They can influence on the adhesion of pathogens, because I didn't bring these results, but we have some results in our lab. We try to mimetize the formation of biofilm using this microbiota, Pseudomonas, Acinetobacter, and Listeria salmonella. You see some protective effect. And this effect we need to study more to have information about. This is a competitive, cooperative, or neutral way to find this kind of results. And we need to thinking about, maybe Christina will talk about the ways to find the problem with the pathogens on the surfaces. But here, my intention, I attended the presentation of our keynote speaker right now, and they talk about the toll of sequencing. We use only the labs at the university, not directly on the industry. Why? In my opinion, we have a lot of different ways to work with microbiota. Our intention is to understand the microbiota and to try to understand specifically kind of industry, how they can influence the pathogens, not the way to change the method of analysis. Because if I try to have information about the Listeria, I can use a method directed to try to find the Listeria. So I need to ask, which is better, knowing the microbiota or finding the target directly? I don't know. We need to thinking about. But I will use the same slide that Dr. John show us. We have a lot of factors that influence microbiota and pathogens in the industry. Raw material that industry works, seasons, temperature. For example, temperature was okay for us with our Pseudomonas results because the low temperature of the rooms when we collect our samples. Our recycling, cleaning practices, organic residues, employees, waste handling, building layout. So it's not easy. If I try to use the same industry, using the same surface, maybe I can find a different results over the year. Okay, we have, now if you can see during IAFP Latino, Gustavo, Emanuele, Márcia, they will present some results that were produced in our lab, and we can talk with them. Raise your hands. Okay. Thank you. Yeah. And I'm finished my presentation, and I need to thank our partners, and of course our research funding, and of course our studies. Without our studies, we produce nothing. In our lab, we have students, they are very important. So I'm finished my presentation. Thank you for your attention. After my presentation, I'm here to answer any questions. Thank you. Muito obrigado.